Perfect. So, yeah, let me introduce myself in brief how I started my career in the organization. So, once I completed my master's program, I joined as a breeder, a drought breeding program in Sriram Bioseeds for 6 months. Then I decided to do my PhD, but somehow I got an opportunity as a support system specialist to support in Corteva Agri Science. So during 2013, I felt this is something different. My what I did in plant breeding something different about the computer applications. So I was a little hesitant to take that role uh, because of some guidance from professors. So I took that challenge. When initially I accepted that role, it was something different in terms of because there is no field work, nothing. So everything I have to uh, do using the computer applications, especially in agriculture sector. You know about the Bayer and Corteva and Syngenta. Mostly these giants, agriculture giants, will have huge impact on data analytics. So I started my career in the system applications. So then I traveled some other countries to get the training in using of the front end applications. So I was there in this role for like 8 years. So I led the Asia Pacific region in terms of the application system support. Then I moved into the core plant breeding as a principal research scientist. But my area of focus will be majorly on the product lifecycle management as well as the data integrity, data management, especially for the region. So recently I took another role called the breeding analyst. So in the breeding analyst, majorly I will be focusing on the machine learning and AI application towards the plant breeding, working on the new phenotyping using the proximal sensing as well as the satellite imaging. So these are my core expertise. My core expertise on the data analytics. So I have like 11 years of experience in the data analytics. So now switch the gear. So uh, when Ravikesh sir contacted me to give some topics on the data analytics, so I'm very passionate to share my experience. What are the recent changes in the analytical pipelines? What are the new statistical methods? How to visualize the data? So for every plant breeder, we are great towards understanding the core plant breeding, how we can do in the field. But that is not the thing as a plant breeder. When I came to the organization, I learned the many things, not only the core plant breeding in the field what we do, but also we need to have some the core analytical knowledge, because then only you can connect the dots, the field knowledge as well as the analytical knowledge. Then you will become like a technical breeder, because there is a lot of difference between the conventional breeder as well as the technical breeder. When you see the three dimension, like molecular knowledge on the molecular breeding, knowledge on the data analytics, and core the field conventional breeding, all these three things will make you as a technical breeder. So these things I learned in the organizations. So so then I will be talking about what are the changes we are seeing in the paradigm shift in the plant breeding, because most of you are aware of these changes, but I will connect the dots how our organization is doing. Okay, and so in the first session, I will be taking you through the changes and how our organization the adopting these changes. Okay, then in the next session, I will be talking more about the advanced statistical methods and how the machine learning can be used in our plant breeding and those aspects. Okay, so that is about. So in the first paradigm shift, everyone knows for every successful breeding program, we need to have the efficient line development. The line development plays very important role successful of any plant breeding program. So in especially in the government sector or private sector especially, each breeder has been given to one breeding program. So for example, these breeding programs are defined by the business teams. For example, I have a requirement in a product like stress drought tolerant hybrid, or I have a requirement product in the coastal segment or something. So these all decisions were driven by the business team. So the business team will communicate the information to the research team, and the research team will convey to the the concerned breeder who is doing the breeding project. So accordingly, he has to develop his germplasm based on the business requirement. He cannot do that. I can do all the germplasm. I can do the recycling. I can make the breed hybrid. That is not the goal. So there is a clear-cut understanding which breeding program has to scatter the products. So Nowadays, we have shipped the gears that earlier we know we are doing like F1, F2, F3. These are like a conventional method of line development as well as we are doing forward breeding. But only challenge is we don't have that much time. We need everything instant. So we need the fixing of the lines. Basically, you are doing this uh, the generation from F1 to F6 because you want to fix the line as well as you are selecting some lines. But you see about the time. Time is very important for us. We cannot wait like more than 6 years or 6 seasons for a fixing a line. So majority of the private sector or even in some CGR institutes, they are using the DH technology. So especially in corn breeding, they are following the DH. So because of this DH technology, we have an opportunity to fix the lines within 2 to 3 seasons. So if you see the next slide, this is the conventional way. But what exactly about DH? I will give you two important points. So in Corteva, especially, every breeding program has an allocation of 10,000 DH lines. It means that a breeder can request to the DH team that he can ask like 10,000 DH lines. For getting the 10,000 DH lines, the breeder has to make the crosses, the breeding crosses. So he can make because he knows what is his breeding program objectives. Accordingly, he can select the parents. He can make the crosses. So, for example, his requirement is 10,000. So, accordingly, he can play the game. For example, he can make the 100 crosses and ask the DH team from this cross, I need the 10,000 100 DH lines. So, 100 into 100, he may get like 10,000 DH lines. So, what the DH team does is, so once the breeding program receives the request to the DH team, the DH guy plants the F1, the breeding crosses, and cross with the inducer. So all the DH, what we are saying, aligns with the inducer. So each organization have their proprietary inducers. These inducers are not common across the industries. So what this inducer does is, because inducer also have the percentage of induction capacity, because currently we are going with nearly 20% of induction rate, because some companies still they report that they are only with 10 to 12% of induction rate, but our induction rate is like 20%. So once they cross with these inducers, the kernels will, you can see like a like white color. Then we transfer those embryos into the Colston Petri plate where the doubling can happens. These what you see here is the DH lines. So all these will be completed in 2 seasons. So if you compare with the conventional cycle with the DH, you see the time. So it's very fast. Within a season, within a year, you are able to get the complete recycling your germplasm to the breeding program. So now every breeder will have 10,000 germplasm, pre-coats. These are pre-coats, nothing but these are fixed lines. So what he will evaluate is, he will evaluate in the top cross, or he may not evaluate 10,000 DH lines. What he will do is, he will evaluate only 4,000 DH lines, remaining 6,000 DH lines he will shelf it, because every DH line is genotyped. So using the genotypic information, we can predict the untested lines. So here we are not wasting the resource. We are using the technology of the genotypic information. Then we are able to predict the untested lines. So this is how we are optimizing our resource as well. And we are able to generate the most of the variability in that breeding program. So for example, we are following this approach not only in corn, we are doing in mustard. So you can see in mustard from the male gametes, we are able to generate the DH lines. And if you see in crops like rice and millet, we are moving towards the speed breeding. Conditions, controlled environment condition, we are able to pass the generation like 3 to 4 generations. So with this, within a 1.5 year, 1.5 year, we are able to fix the lines. This is how we are rapidly increasing our line development in each breeding program. So now breeder will have efficient germplasm so that he can evaluate as per wish. So he can make the some selections within the pre-coats and evaluate in TC2, then come and do the predictions, then select the untested lines, directly jump into next hybrid evaluation. So this is all available because we have ample of the fixed lines. If you don't have these fixed lines, you have to wait like more than like 2 to 3 years for getting the fixed lines. So this is a complete shift in the line development. Now coming to the next shift is using of the marker information. I think most of you are familiar because most of your thesis or your research work contains this molecular breeding work, especially. So when you compare during the early generations, because genotyping is very high efficient in cost, but doing the phenotyping is cheap. Now what happened is the genotyping cost become less and the phenotyping cost become huge, because the scarcity of labors, and also we are going to phenotype huge number of lines or hybrids, so it become more laborious and more resource. Rather than doing genotyping is very easy. So using this genotypic information and all these the as I said, the phenotyping is very expensive and genotyping become very affordable. So we used this genotyping information. We are so this is the, I think, very clear from where we started and how we are moving. So then you can see now we shifted to the Jivas. So I think most of the people are aware of this marker trait associations and the whole genome predictions. So I will talk about this how we are using in Corteva. So this is especially if you have any like even you can do in the forward breeding, or if you have the marker information, it is very easy to crack the identifying those trait and screen the susceptible entries. But if you do the whole genome prediction, this is where we are doing majorly focusing. Here what we are doing is we used to do the robust predictions. For example, we are not evaluating hybrid at initial stage. So what we are doing is we develop the estimation sets, we develop the training set based on the genotypic information as well as the phenotyping information. Then we used to predict the hybrids. We may say, hey, I want to cross this A into C. What will be my predicted yield? So like this, we used to play. So we will not evaluate at initial stage. We use this marker information, high throughput phenotyping, all this information. We used to directly predict the hybrids. So because of this, we are not testing at initial top cross to hybrid evaluation. We are directly jumping the hybrid at higher level of testing. So because of this, we are saving the lot of resource and we are able to screen more hybrids. So this is what we develop the training population, we genotype and train the genomic selection model. Then directly we can calculate the genome estimated breeding values. Because if you see only the phenotypic information, we are not confident about how the hybrid is performing. We are including the marker effects as well. So we are generating the genome estimated breeding values. So that makes us robust selections. So because of this, we are able to fasten our cycle. So for example, you can see we genotypically subset the samples, sorry, and we have the phenotypic information. So we combine both, we reject all this portion, we select only the cream. So this gives more value to the organization in terms of the resource optimization as well. So this is one of the major shift, and we focus more on this creation of training set, estimation sets, and based on that we select the hybrids. So just wanted to show you how our line development happens. So we have clear cut. If you see this video, small video, how we are differentiating the heterotic pools, the male pool as well as the female pool. All this information is derived from the genotyping. So if you see this small video, you can see this side what you see the female heterotic pool, SS stiff stock, and what you see this side is the non-stiff stock. So we have the clear-cut demarcation between the heterotic pools. So when you have this clear-cut demarcation, it is easy for you to make the crosses or hybrid evaluation. What you see here is the maturity of the lines. So we have from 75 maturity to 120. This is a male side, this is a female side, and this is a male side. And what you see here is which year these inbreds were coded, which year inbreds were fixed. That is what you can see this information. So we have all this information in the database. Any breeder have in the Corteva, they have access to these applications. They can pull the data, and if he find that this germplasm will work in their breeding program, he can happily use those inbreds and make the crosses. and subsequently he can recycle the germplasm as well. So we have sophisticated platforms, and all this information is available in the database. And one more important thing I want to emphasize is, I'm not sure how many people are aware about the breeding funnel concept. So here, as a breeder, or we need to shorten the length of the our breeding cycle and widen the our breeding funnel. It means we need to we need to test more combinations, and we have to shorten the cycle so that we are able to identify the product in short time. If you increase the length, like development of the lines, hybrid evaluation, step 1, step 2, step 3, it takes many number of years for a commercial release. So what I am doing is, as I said, using the DH technology or speed breeding, we are able to test the more recombinants, and we are by using the predictions, what I talked, the genomic selection, whole genome prediction, we can directly jump the hybrids. So we are shortening the testing time. We are increasing the widening the cycle for more recombinants. We are using the whole genome predictions to identify the right hybrids and directly jumping. So we are reducing the length of the breeding funnel. So it means we are our product life cycle is hardly 3 to 4 years. This is what I was saying. The product life cycle management is, so at every 4 to 5 years, we have to replace the new hybrid. That is a product life cycle management. If you are not successfully delivering the commercial product within 3 to 4 years, you are not successfully running the breeding program. There is some challenge in your breeding program. Why commercial hybrids were not coming up? So my role is, I have to identify those gaps in each breeding program, either in terms of the germplasm or in terms of the strategy. I individually work with each breeder along and assess the breeding program. So assessing the germplasm, what is the challenge with the parents, how to bring the external germplasm into their breeding programs. So that is what my role, working with the breeding program, identifying the gaps, fill the gaps, so and develop the germplasm fast. So another another paradigm shift is using the digital and high throughput, as well as using the advanced statistical methods. So this is where I spent like more than 80 years, especially in the new phenotyping areas. So if you see this image, which clearly says how in earlier days we are planting. So now we are having the sophisticated GPS enabled. What I was talking, GPS enabled. There is a driverless. So this tractor is GPS enabled and no driver is present. So automatically all this information we capture in the database. The moment we give the signal, this tractor will automatically plant. We give all the data, how many rows it has to plant, what is the length of the row, and what is the alley width. Every information we give in the database, and we just pass that information to this GPS enabled tractor. Automatically, this planter will plant. So there is no human interference here. So how we are shifting manual, how much laborious, here no human interference is available. Directly this tractor will plant. Now we are in doing the phenotyping. For example, this image, particularly we are evaluating the lodging in corn. So the person goes to the field and you see how many plants were lodged. So whether how much wind force is there, all these parameters he consider. Now we have sophisticated mission called Boreas mission. It pumps the high wind to the field. So then it can estimate whether this plant able to lodge or not. So that is how it calculates. And if you see this, in earlier days, we are writing the phenotypic information in the book, then translating into Excel. So now we have a sophisticated combines. So what it does is, it individually harvests the each plot or hybrid, and it calculates the what is the harvest weight and what is the moisture in that grain, and directly send this information to the cloud. So here, nothing, no human interference. There is no book and paper, nothing. So automatically Vision goes, harvest, calculate the harvest weight and moisture information, send data to the cloud. So nowhere there is no human interference. So we advanced this much technology. Now coming to the using of statistical methods. So here you can see this field is having some challenge, and this field is having some tilt, and there is a there is a border effect basically. So there are a lot of challenges in the field. When you see this kind of whenever you plant this experiment, when you find this kind of challenge, when you try to do analysis, there will be always bias in the analysis. So because of these challenges, how to nullify these challenges? So I think if you are aware the blues and blobs concept, so it is similar that the spatial adjustments. In Corteva, we majorly rely on this because we don't replicate our experiment. Because as I said, we have to reduce the resource. So what we are doing is we are going the spatial design. For example, each color indicates one experiment. There are 6 experiments were planted in this location. But what you see the diagonal checks, diagonal information, these are repeated checks. So what we are doing is we nominate 4 stable checks. Means stable check means these are pioneer stable checks. It means that these 4 checks will be repeated in a diagonal fashion. Okay. So by using this mixed model analysis, mixed model things, we are able to calculate the noise in this location. Earlier, we are able to we used to replicate replication one, replication two, block one, block two. So it is consumes lot of resource. So by using this advanced statistical methods, we are able to calculate the same amount of information when you go replication. Even without replication, I can able to calculate that noise in the location. So I can able to calculate the heritability of the location and all those information. So if you see this is a raw data and this is adjusted data, you can see this density curve. So it clearly says the unadjusted data, there is a lot of variation. But once you do this mixed model fitting, we are able to adjust the data because I have seen this. I have compared with the spatial effects, without spatial effects, literally on the field, because some entries I wantedly kept where you have more fertility gradient, and some entries I kept where there is some patch. When I do analysis, I see some challenge. But when I used this mixed model analysis, when I fit this model, I was able to adjust the values. So that is the real value. That is the best linear unbiased estimates. Because when I calculate at location level, it is the estimate. When I calculate across the location, I was able to estimate the predictions. So I think nowadays, I hope many people, you guys, the young plant breeders, using this kind of mixed model analysis in your thesis, that gives a lot of input. Definitely, you will see there is a huge difference between uh, when you analyze with the average model versus the mixed model analysis. You will see a lot of impact in your hybrid selection as well. So this is what I was saying. When you see the scatter, the raw data location wise, location 1 versus location 4, the same set was planted. You see the data was scattered. But when I fit the model, mixed model, you can see the adjusted data. Mostly it was not scattered. It was aligned. So there is a lot of value when you include these kind of sophisticated advanced statistical tools in your analysis. So this is what I was showing in the earlier slide. This is the sophisticated Boreas mission to estimate the lodging tendency in the corn hybrids. Especially we do in North America to estimate the lodging tendency in corn hybrids. This mission pumps high velocity 100 miles per hour the wind. So then it can estimate whether the corn stalk is lodging or not lodging. So accordingly it records the data. So this is very fast and sophisticated information. So based on this, we used to generate the breeding values for each parents as well. Then coming to as I said, I was expertise in the proximal imaging. So this is the mission in our rice grain quality lab. When you send the sample of rice milled grain, it will able to estimate the chalkiness as well as the translucent content immediately. Just it will take the image and it able to generate you the translucent and the chalkiness, the opaqueness, everything. And recently, we developed a model. So myself and my team member, so we developed a model that is using the deep learning. So what we are doing here is, in the paddy, you can see open husk as well as choppy grains. So it is creates a lot of challenge in hybrid advancement. Even in the miller is not interested to see the choppy information or open husk. So how we can estimate? Because counting the openness in a given paddy sample or counting the choppiness in the given paddy sample, it is laborious and cumbersome. So we thought using the proximal imaging can we do immediately or in a fast manner. So we developed an algorithm using YOLO. So what it does is when you given a sample of like 1,000 paddy grains, and it takes the image and it can tell you how much percentage of open husk and how much percentage of choppiness in this given sample. So this is what this is recently, like last month we developed the model. You can clearly see the filled grains, choppy grains and open husk grains. So you can see the full grains, choppy grains and the open husk. So the model predicts and gives in an Excel file. So this is what we have to use this technology for the rapid improvement. And coming to the drones, I'm not sure. So I have expertise in analyzing the drone images. So 3 years back, we fly the drones and we capture all the images in the field, especially in the field, especially in the corn field. So then it takes the photographs. So then I used to stitch the image, then do the plot clipping, then able to extract the information. For example, you want to count how many plants were in each given row. So, or you want to estimate the plant height. So what we do is we fly the drone, get the information, then process the analysis. Then it generates the information. So it's very easy and very fast. So a person can go to the field, fly the drone, send the image, then run the analysis. It will generate the huge information. So sometimes human makes a mistake, but if mission makes the mistake, it is common across all the plots. So we can find that biasness and we can reduce that biasness. So this is one. And also we are working on the satellite imaging. So I did not put because recently we started that work. So our objective of using satellite imaging is recently we did estimated the soil moisture using the satellite. For example, if you have some GPS coordinates, if you give me the GPS coordinates, we are able to estimate the how much soil moisture present in 1 meter of depth using satellite imaging. And our next project we are working on identifying the hybrids. For example, if you have some flagship hybrids, you want to estimate how much area this hybrid is coverage in India. So it is very difficult for you to estimate. But if you develop a model using satellite image, you are able to estimate approximately this much area is covered by this hybrid. So what we are doing in that project is we are estimating the for each hybrid there is a unique signatures. So we are able to capture those signatures, put in the model, develop the model. Then once we have the satellite coverage imaging, then run the model, it is able to estimate this much area is present with this hybrid coverage. So this is most advanced technology. We are just started in India as well. So just wanted to show this is the first drone we started in India. This is MJ600. So I just told this is a GPS. This is very important in any drone fly because it is the thing, it will generate the GPS information for each plant level. So yeah. And as I said, the cloud computing, whatever the data we collect, it will go to the cloud. From cloud, we are able to access the data. There is no repository. Directly, whatever the data, it goes to the cloud, then go to the cluster, centralized cluster, high throughput cluster, where the processing analysis will happen and generate the results. So it will be some real-time applications, some static, some dynamic, all things can be done in the fast manner. So I think the next step, next shift is about the value added traits. I think you are familiar about the biofortification, something like that. So similar way we are focusing, especially in North America. We are not doing anything in Asia Pacific region, but especially in North America, we are doing on the soybean. So we have the high oleic acid. So we have exclusively enlist trait, the transgenic trait. So where we have the high oleic acid content was improved in the soybean, and we are branding as high plenish. So we are not selling as a normal soybean hybrid seed. So we are marketing as a high plenish. Plenish is nothing but the trait what it was involved in this hybrid. So we are marketing. So something like this we can do in especially in mustard. So we have some issue with the mustard brown rape, the Warabank issue. So we are working on that, and especially in mustard we have sclerotinia issue. So we have some traits, native traits we are working, or especially in rice we are moving towards the direct seeded rice. But the challenge with the direct seeded rice is about the weeds. So we are working on the HT hybrid tolerance, which is a native trait. So there are some projects ongoing. So especially we are working on that area as well. And the other important shift is the disruptive technologies. So I told you the just now one disruptive technology about using the satellite imaging. So because drone is a proximal imaging, but satellite you can get the more information. But the challenge with the satellite imaging is the cloud coverage and what kind of information you are getting and what kind of satellite, because in satellite there are like RGB cameras, hyperspectral, multispectral, there are lidar, there are different sensors. So the challenge with this satellite imaging is the money. So we need to spend a lot of money getting those kind of images. How much, what kind of information you want, at what level, pixel level you want, like 10-meter resolution or 50-meter resolution. So all this based on your project objective, you have to get that information. So one of the descriptive technology you may aware about the CRISPR-Cas. So it was a proprietary to the pioneer, I mean Corteva. So we developed in the Vaxicon. So this is one of the technology we used, and it was local. I mean, it was developed in North America. And I know how. So we are. I mean, I am talking about in Norton Corteva. So how the technology, the disruptive technology is using in the agriculture sector. For example, you can grow the meat in the lab. You can define what kind of things. So the first burger was cultured, which was sold these many dollars. So this is what the impact. What I was talking the future. So just wanted to show you how we in Corteva we work. So we can, as I showed you earlier, the clear-cut demarcation between the two heterotic pools, the strip stock and non-strip stock. So clearly we demarcate, because any successful breeding program or anything you want, you need to define your heterotic pools clearly. Then only you can play the game. You can make the develop the breeding process within the heterotic pool, and you can make the cross across the heterotic pools. So all depends how efficiently you are able to demarcate your heterotic pools. So this is the inbit development. What I talked about, the DH technology, evaluation, all the line development, everything happens here. And directly we jump into the hybrid evaluation. So you can ask what is R1, R2, R3? This is a stage, T1 stage, T2 stage. The only difference what you can see is each stage, when you are going step by step, the in number will be more, number of locations will be less. When you come down, your number of hybrids will be less, number of locations will be more. That is the logic. That is the size what you can see. In each year, coming to when the hybrid was, when you, for example, if you are evaluating like 200 hybrids in 10 location, your selection intensity is only 5%. Then you advance to the next stage, and instead of 10 location, you will be testing 20 location. Then again your selection to 5%. Then you will be testing here 40 locations. Then you send it to the business team, and business will evaluate in 100 locations. So this is the way of testing we do. It's not just like a few locations. We combine like 2 to 3 years of locations, and we calculate the year effect, and based on that we used to advance the product. And simultaneously, some breeding programs in India we don't have chance to use the transgenics, but in other countries we immediately, once the line is coded, we immediately start with the trait conversions, because each breeding program we, based on that, we used to transgene the lines, though simultaneously the trait integration program will start once the line is done. So they has to generate the transgenic coded line at R3 stage. So this is parallelly, once the breeder codes the lines, it fixes the inbreds. Simultaneously, the trait integration program starts, and the team will convert the lines according to the transgene, then deliver. And other thing was, I was talking about the genetic gain. So every year I used to sit with each breeding program and calculate the genetic gain. How much? For example, I took only example of yield. So I used to calculate for yield, moisture, year height in corn, in rice, the flowering. There are different crop, different objective, different trait. So so what you see each year, you have the 10 years I took. What you see in the bracket is how many hybrids it was tested in each year. So, for example, you see this particular breeding program is making 0.17 metric tons genetic gain year on year, because 1 year you cannot bump. It is a cumulative effect. So, for example, there is a dip in this year. So, I can ask, have you changed any germplasm, or there is a challenge, any challenge like high rainfall, high stress here. That is the reason your hybrids were not performing. Why there is a sudden jump in this year, the performance of hybrids? So all this information we used to collect, and we used to discuss, take the collective decisions. Once you fix these things, you can see every year there is no up and down. So we fixed that. What is the challenge? Whether the challenge with the geography, whether challenge with the germplasm, we are able to figure out. Then you can see next year onwards, we are seamlessly increasing our genetic gain. We are recycling the lines, we are testing, recycling, testing. So we are able to increase. This is the uh, success of the breeding program or a breeder. So he has to recycle the germplasm, deliver the right product to the business. That is the objective. So I used to calculate this genetic gain for all the crops, all the traits, and I identify the gaps, and I used to help them fixing those gaps. So if you see now, you can compare. I was just showcasing you how the Corteva, the breeding systems. So especially in Corteva, we have a very sophisticated platforms for each and every step. For example, if you want to set up an experiment, what kind of design you want to use, there is sophisticated platform. All these applications were in-house. It means there is a dedicated software team sitting in India as well as in North America. They developed applications. All these applications are in-house. It means it is not available publicly. So all the data is present in the repository. So a breeder can open any application, he can pull the data, fetch the data, and he can say, I want to create experiment. Automatically, it create an experiment. It maps the experiment, and automatically using the mobile device, he can collect the data, send the data to the database. Then all the data quality process will happen behind the scenes. Then submit the jobs, then get the data, the blues and blubs, do the product advancements. Everything is seamless. There is no human interference. Not collecting the data in Excel, typing the data, doing analysis, nothing. Everything is sophisticated platforms. All this was developed by in-house. So every year we used to enhance the applications. For example, you can see this is what I was talking the earlier breeding funnel. So how we moved. So it is about the genotyping information. We have the all the genetic map for each crop. Then this is what we talked earlier. This is about the heterotic pool information. This is about the robust pipeline. So all these breeding systems, I was just taken very glance because as per the policy, I cannot reveal a lot of in-house applications. But I just taken you few approved slides. These are the sophisticated breeding systems. So as I said, cross, do the top cross, code, fix the lines. This is the objective. You have to recycle the germplasm as soon as fast. So as I said earlier, the initial level of testing, what we used to do, how many locations, as I said, the conversion process will start. Once we move to the next season R2, R3, then the business team will come at R4. Then they evaluate in the large number of locations. Then simultaneously, it is not about hybrid evaluation. You have to take care of your parents also. Because once it is commercialized, you need to get the parents also increase the seed. So simultaneously, if the business says that this hybrid is performing well, simultaneously the parents of the hybrids will be parallelly increased by another department. Then once you take all the cumulative decision, once the business, research, agronomist, everyone will sit together in the decision meetings, assess the data quality, assess the hybrid performance. Then we give the signal that let's commercialize this hybrid. That is how you see the new product in the market. So it is not that there is a huge amount of time, resource, everything is spent for releasing of single commercial product. As I said, from 10,000 DH lines, you are able to deliver single commercial product. So that much amount of resource we are able to spend for getting one. Now you can see the breeding, how we are doing earlier, how we want to do now. So select the best, cross the best, hope the best. But now you have to select the best, cross the bet, and you have to create outstanding commercial product. That is our objective in current scenario. We should not hope. We have to create. And I just wanted to give the what because I studied in the university, then I moved to the commercial organization. I just wanted to give what are the gaps, how the public and the private commercial organization, I would say commercial, not the private. So here everything is matters. If I say this much resource, what I am going to deliver. So everything is calculated in the commercial sector. So commercial organization. So especially if you see the what I was learned actually when I moved to the organization. So the programming language and the data science skills. Because if you say I'm just a conventional breeder, I can do the best cross. I have the breeder eye. I can select the lines in the field. It doesn't matter now. It can be done through sophisticated platforms. But what you need is you need to know some data analytics, because these data analytics play very important role. If you have the domain expertise and if you are able to analyze the data by yourself, that adds a lot of value. Because if you say I know the only conventional breeding, but I am unable to write the code, or I'm unable to develop an estimation set through using some modeling, or I don't know machine learning, so it is very difficult because you have to relay with other people. So you need to explain what you need. So rather than if you have these sophisticated soft skills and if you have the domain expertise, it will add a lot of value in any commercial organization. So now commercial organization is looking these kind of graduates actually. They are not looking that I did their thesis work on this. They are not looking in that areas. They are asking what extra you can bring, because they are all expertise, because these are commercial organizations, they have all the domain expertise, but they are looking what you can bring to the organization, because all the platforms were established, when to select, what percentage you have to select, everything was written. It is all about then what you bring to the organization. What I what i wanted to give message to the upcoming graduates: try to learn some programming language. Try to start initial, like at least, because why I selected on R and Python, these are publicly available, no need to pay any money. Just start learning or take your data, try to analyze, try to visualize the data. Nowadays visualization also plays very critical crucial role, because you are doing a lot of analysis, but the visualization will communicate the right information to the audience. If you are not making the right visual, it will not able to communicate to the right audience. So so you have to learn, like start learning R, Python. I also started when I joined this organization. I know little bit about R, but I'm not familiar about some other programming languages. Then I started when I enrolled the course in Reply to Hyderabad. The platform they used is Python, so it is mandate for me to learn the Python. So I started learning Python. Then I used to write my own codes and able to develop the algorithm also. So it's very simple. I think in the next coming sessions I will show you uh, what is this machine learning, how you can connect, how you can bring your data, and how you can train the model, and how you can predict. So nowadays you are hearing like more predictive breeding, not the conventional predictive breeding. How you can predict using the marker information, phenotyping information, how you can predict. So try to learn some programming language, try to learn some basics of machine learning, which I will be covering in the next sessions. How to focus, what are the areas, uh, uh, what are the freely available courses are available in some Udemy courses or some Coursera, where you can enroll, and they will have some data sets, and you can try to code, and you can play with your data. You can because you are generating huge data with your research programs. Try to play with your data, how you can visualize. These are the things, these are the gaps I have seen, especially when I interact with the people, especially in the agriculture sector. They are very expertise in the field knowledge and the conventional, but when you talk about the sophisticated, like advanced statistical methods and the data science skills, this is the lacuna I see. So so please focus on these areas and try to fill the gaps. At least I'm not telling to become you like expert. At least have some basic sense, basic knowledge about these skills. Definitely, when you attend some interviews, definitely these things they are emphasizing more, not on the pure the research work what you did and what conventional knowledge you have.